Okay, then we can continue with more pictures and less text on slides to discuss the neural origins of EEG signals. And as you will soon notice, the theory or the basic model about what the neural origins of EEG are is fairly straightforward, fairly simple actually, which is, I find it always a little bit surprising because, for example, if you compare it to fMRI, where the bold signal is a complicated thing and so on, and it's not really understood actually, it feels like the EEG signal is very well understood in a way. But actually, I think that what's actually happening is that there has been less research on the origins of the EEG, and definitely most of the EEG research that is being done right now is more either as a biomarker, so in a clinical setting, so does it tell us something about disease, or as a cognitive process marker in cognitive neuroscience. But there's, I would say, hardly any work on understanding the EEG signal per se. And I would guess if there would have been more research, or would be currently more research, that things would be a bit more complex than the story that I'm going to tell you now. On the other hand, it's good because the story is straightforward. One feels that one really understands this and everybody's happy. And that's also not too bad if people are happy. So, good. To talk about the neural origins of the EEG, of course, we need to first start to zoom in and, of course, have a look at the system under study, which is, of course, the brain. So here, a human brain, where there are two main tissues, namely gray matter, and specifically the gray matter forming the cortex, so the outer sheet of the brain, and then there's white matter, which refers to the axon bundles. So as you all know, the gray matter holds the cell bodies. Some neuroanatomy and some revision of that will be done in the cognitive neuroscience courses. If you don't feel like you or these terms, you are not that familiar with these terms anymore, there will be some revision of neuroanatomy in cognitive neuroscience. Here, I just want to emphasize that you should think about the thing called cortex. And actually, most of cognitive neuroscience, besides the things that are related to the basal ganglia, is actually work on the cortex and the mapping of cortical function. So the cortex is an important thing in cognitive neuroscience. So then zooming in a little bit more, so I assume that you have also come across this notion of Brodmann areas, which essentially say that the cortex is not uniform in terms of its histology. So if one basically cuts up a brain and then puts the brain slices under a microscope and looks at the histological picture, for example here using a staining method that mainly stains the cell bodies, or these selected cell bodies, then Brodmann already 120 years ago found that it's not that the cortex histology differs across the cortex, and this then gave rise to this idea of Brodmann areas, which are areas of cortex which are relatively uniform in terms of their histology. And these Brodmann areas still sometimes come up. So, for example, area 17 is visual cortex, which is shown here, is still something that people talk about. one thing to note, for example, about these differences in histology is that here primary visual cortex, where which refers to the first cortical area where visual input is processed, has a relatively large layer 4, which is where the input comes in, whereas, for example, something like primary motor cortex, which actually holds the cell bodies of the cells that send out essentially the muscle activation information. So it's mainly an output area, has a very small layer 4. So these are kind of the histological differences in the extremes that have been described in terms of these Brodmann areas. Actually, by now, of course, Brodmann area, this is histology like 120 years ago. There is still, of course, active research on basically parcellating cortex according to certain functional criteria, certain connectivity areas, and maybe even together with some macro or mesoscopic views. So, for example, the work that they do in Jülich, so the Karl Zillis, Katrin Amunds, and the guy who developed the anatomy books for SBM, whose name I'm currently not recalling, they still try to identify different areas based on different criteria of the brain. That's all very nice. I think it doesn't really help to understand how the brain does cognitive processes, but of course it's also very important from describing the brain, and also then linking different brain areas with, for example, certain pathologies. Hello, can we go on? Yeah. So, as I said, these Brodmann areas are based on different kind of histological stains, and what this then shows is the typical 6-layer makeup of the cortex, where the layers are numbered from 1 at the pial surface, which is going to the pia, which is one of the sheets of the cortex, and layer 6 is the lowest layer that borders to white matter. So this is outside and this is inside, if you want. And these 6 layers have names. So molecular layer, which doesn't show any thing if you do, for example, a Golgi stain, external granule cell layer where you have small cells that are thought to be interneurons, and then the external and internal pyramidal cell layer, and these, the pyramidal cells will feature quite a lot. So they have their cell bodies here in layer 5. And they project axons to the white matter and have their apical dendrites here. Here you can see that again. So the cell bodies of the pyramidal cell neurons lying in area 5 for the internal pyramidal cells. There are also external pyramidal cells, but we'll be later on mainly be interested in this internal pyramidal cells. Here you see just different stainings. Golgi staining selected cell bodies. No one really knows which ones and why. Lizard staining, and not only cell bodies, but also the axons, so the axons and dendrites. Dendrites and axons. Here the cell bodies stained by Nissl stain, and then you can also stain, if you want, the protrusions of these cells, so axons and dendrites, using a Weigert stain. More in terms of the connectivity, and also what I already mentioned. So the fundamental, or a very simplified idea about cortex, or a fundamental idea in terms of simplified, is that input is reaching a cortical area in layer 4, where you find, or which is also referred to as the internal granule cell layer. So there are cells, they are not the biggest cells, and they are thought to be interneurons. But importantly, there also are many dendrites, so you can have synaptic connections. And then layer 5, holding the cell bodies of these pyramidal cells, which project their axons somewhere else, then being the main output layer. This again is in line with what I showed you earlier with the primary visual cortex, which has a thick layer 4 because it's mainly an input area, and the motor cortex, which is a thick layer 5, which is because it's mainly an output layer. And then these other layers, they do some local processing. What that means in detail and so on, this of course depends always a lot on whether you actually model that, what kind of things you model and so on. And it's actually something that, depending on the analysis method that you use, for example for EEG or for fMRI, can maybe play a role, but in standard data analysis techniques usually doesn't play much of a role what this local processing really means. There is some nomenclature that you might come around when reading on anatomy. So these, in the white matter, the axons that come from a given area here, they are referred to, for example, as cortical. I make this a little bit bigger, sorry. So there's this term cortical or corticothalamic, corticoclaustral, which means it originates in the cortex, so cortical, and then it goes to some other cortex area, or it goes to the thalamus. So that's just this nomenclature here. And the general idea is that you have, in terms of the cortical connections, so now I'm not talking about the output of, for example, the motor cortex to the spine and then the muscles, but in terms of cortical connections, so connections that originate, so axons that originate in cortex and go to other cortical areas. The idea is that ascending connections, so going from sensory more low-level areas, originate in layers 1 to 3, and then again get into the input layer 4 in a higher cortical area in terms of a cortical hierarchy, while there are also cortical connections that originate in layer 5 and then end up in many layers. Yeah, so this is just what this describes as kind of the wiring of connections within the cortex. And of course, all these descriptions are the result of tracing studies and histological studies. And of course, if you look closely and different areas and so on, then things get more complex. But by and large, averaged over many studies, if you want, this seems to be one of the kind of fundamental connectivity motifs in terms of axons that originate from cortex layers and go to other cortex layers. And these kind of things are then sometimes mirrored. So from my perspective, dynamic causal modeling for EEG or delayed differential equation modeling for EEG, which is something that we published 2 years ago, 2017, which is essentially just a review of dynamic causal modeling for EEG. This is the only imaging techniques that actually is interested in these things and also models them. So happens in EEG, these kind of things, but if for example this afternoon, if you hear the EEG talks this afternoon, there will be no talk about connections in cortex. Okay. Zooming in even further from this kind of mesoscopic and connectivity motif level to the cells, then of course we are again at something that you are very familiar with. So the typical neuron, and we are now interested in these typical pyramidal neurons, has a cell body, it has its apical dendrites where it receives its input, and it has this axon which, for example, a pyramidal neuron in motor cortex would project to the spine, but of course you also have a lot of pyramidal cells in cortex that don't project to motor cortex but to other cortex areas. Yeah. And then this actually, there was something I wanted to say. No, I forgot. But then the axon goes to another cortical area. And this is always the kind of the textbook representation of neurons in Niedermeyer's EEG. I think it's a little bit more realistic if that can be said of something like this here. But what it shows is that the neuron, of course, makes many synaptic connections, and then it doesn't just float there in vacuum space, but there are a lot of glia cells around, and everything is squished together and kind of mushy. It doesn't look like as nice as this here. So why are we talking about this? Because I first of all want to remind you of that, and we also now, from again knowing what a neuron is, want to now talk about how how electrical activity that happens at the neuron level relates to the EEG, which of course brings us back to something that you have maybe heard in school, or at least in psychology, action potential. So who has never heard the term action potential? The other way around, who has heard the term action potential? That's all of you, and that's kind of what I expected, and which is quite clear. So, does anyone want to explain action potentials then, as you all heard about it? And I don't have to explain it. No one? It's tough.
So a signal comes in from an axon terminal onto the dendrite, which depolarizes the membrane to minus 55 millivolts, and then sodium comes into the cell and depolarizes the next part of the membrane, and this way the action potential travels across the axon until it reaches an axon terminal and maybe elicits an action potential on other cells.
Yeah, that's the basic action potential story. So very good. So I don't have anything to add really. One thing that is kind of interest is that the resting potential is negative and is kept up with using energy. So you know that there are these sodium-potassium pumps, so that this resting polarization always is there. And just as you said, EPSPs can be elicited in the dendrites, and then if a certain threshold, and you had, I think, the 55 millivolt, minus 55 millivolt threshold is crossed, then you get this massive influx of positive ions and this peak, which then, as you also said, I just repeat everything, can travel along the axon. The important thing here, and also from what we will talk about later, is that all these measurements that you see here, so the EPSP activity, for example, in this dendrite, or the inhibitory presynaptic activity in this one, and also, of course, the action potential, this is always measured with an electrode put into the cell, right? So it's the negativity of the interior of a cell with respect to the outside. And And these kind of preparations, of course, you can do in vitro. For example, of course, the most famous studies of this are the Hodgkin-Huxley studies in the 50s, where they used the giant squid axon to basically establish this traveling of action potentials. So these are kind of in vitro studies, and of course, we don't put electrodes into cells when we do EEG. However, these processes, and specifically the EPSPs and the IPSPs that are evoked in postsynaptic cells, they play an important role for what we actually measure with the EEG. So this slide here shows essentially a cell body of a postsynaptic neuron here, for example, at a dendritic site, and here you have some other neuron sending its axon, and you have this presynaptic terminal. So of course this is then the synapse, and if there's some excitatory activity, then from the outside positivity must flow in. So you have a net decrease in positivity at the site of the pre-synapse, because positivity is going in. And this means that if you're now looking at the outside, not about the action potential, but you're looking at the outside of the cell, then there is actually a decrease of positivity here, which means there is a potential between some parts of the neuron, because here you suddenly have less positivity, which means here you have more positivity, which induces, which then has a potentials and induces a current flow. This shows the reverse. So if you have something that is inhibitory, then at the site of the presynapse outside, you have an increase in positivity, which induces a flow then of positivity away from this. Yeah? So what this slide tries to show is that when these EPSP and IPSP processes happening at the level of the cell projecting to another cell, some things also happen in terms of the flow of ions in the extracellular and, but this is not that important, at the intracellular level on a bigger scale, kind of a, not really mesoscopic, but at a bigger scale. And we will look at that again in more detail, but for the moment, the stuff that happens extracellularly, in extracellular space, is, of course, it's not only one synaptic site that is active, but there are many synaptic sites, and of course there are also things like action potentials, and all of these things, they generate extracellular currents, and these extracellular currents and their associated potentials are referred to as field potentials. So if you put an electrode into the brain extracellular, so we're not talking about action potentials now, but we just put it somewhere in the vicinity of neurons, you put this electrode there, and you have a reference site, then you can say, okay, this is where the electrode is this much more negative than or positive, depending on what's currently going on, than some reference site. And this is the potential that is then registered by this electrode. And the point of this slide is that this potential that you can measure there, depending on how far essentially you measure that away from the neuron or neurons of the cortex, has different names. So if you measure these potentials at the skull, then this is referred to as the electroencephalogram, so EEG, what we're currently talking about. If you measure these potentials at the cortex, so you basically have to think that you take away the skull and put an electrode directly on the cortex, which sometimes happens in epilepsy patients, for example, for clinical mapping, then this is referred to as electrocorticogram. And if you actually put the electrode very close to the neuron, so you essentially poke it into the cortex, then these potentials are called local field potentials. So the point is that if you read about local field potentials in electrophysiology, this is the same activity as recorded as the scalp. But of course, the skull and the intervening tissues act as additional filters and essentially low-pass filters on these potentials. So as we will see on the next slide, the amplitudes are lower and more. So, but you should not think that EEG and LFP are kind of different kinds of refer to different kinds of neural activity. They all refer to these extracellular potentials and currents that occur because these interneuronal interactions at the synapse take place. The thing about the term local field potential, which is an electrophysiological term, is that the term is just wrong. So it has nothing to do with it has as much to do with an electrical field as any potential has to do with an electrical field. As we discussed earlier, it's just a potential. It's just like you put an electrode on the skull and you measure the potential with respect to a reference site. The local field potential in the cortex is just the potential with respect to some reference site. And the term is just weird and doesn't make any sense from the physics theory of an electrical field and an electrical potential. And again, the same activity that is due to these cellular and intercellular processes. Of course, at the same time, like any electrical activity, evokes or has a magnetic field, and this then measured is the MEG. This we will talk about. So this is trying again to drive this message home. So here you have examples for recordings inside brain tissue where you have some electrodes inside the brain here of a patient for epilepsy mapping. Here you don't see the electrodes, but you can put the electrodes in here of macaque monkey. Here the electrocorticogram where you put the electrodes on the cortex, and here example of MEG again. Just to make clear that this is always referring to the same underlying neural activity but measured at different distances. And this, all these last slides, they are actually from this review by Bouchaki, who's a very famous EEG person, from 2012, Nature Reviews Neuroscience. I don't think it's actually in the references, so maybe just Google it, and I put it in there if I don't forget. This kind of makes this point that this always refers to the same activity. So this here shows simultaneous recordings at sites in a patients with drug-resistant epilepsy, hence the depth electrodes, which we call LFP, the ECOG electrodes, which are applied to the cortex, and then scalp EEG. And the main point here is that this is not congruent in time or anything, but what it wants to show is that that the amplitudes are larger if you have the electrode closer to the origin of the activity, which makes sense because things get attenuated as you go further away. And a little bit, although that's really not that easy to see here, is that you also have higher frequency patterns this is not really convincingly shown here. I mean, this looks a little bit like, so here you have something rather low frequency in the ECOG and you have higher frequency here. But yeah, the idea that, so you know, when there's a party somewhere and you go into the party, what you hear from far away is mainly the bass. So and as you go closer, you also hear the higher frequencies. And the idea is that the same thing happens here, so that you have high frequencies that you can observe with LFPs and ECOG data, but then it gets attenuated in the EEG and the ECOG. That's the idea, but of course it also depends on with which kind of temporal resolution you actually image and so on. But yeah, so the main point being that it's always the activity that we now want to discuss once again in more detail, that you can register with electrodes very close to neurons, a little bit further away from neurons at the cortical surface or at the scalp. But it's not kind of that at the scalp you measure the action potential, and if you put the electrode into the brain, you measure the EPSP or something like this. Yeah, so it's always this summed extracellular potentials in current flow that arises from the fact that synaptic activity happens. So again, about this extracellular current flows, so how they are induced. So here you have the resting state of the axon, where you have some negativity inside, so you have this resting potential, you have positivity outside. Then the synapse gets activated, you have in the EPSP process you have positivity flowing in, meaning that along the membrane, because positivity here is flowing in, along the membrane here outside, you have a surplus of positivity here and of negativity at the location of the synapse, which means you have potential between this part of extracellular space and this part of extracellular space, which means then you will also have a current flow here, and the difference between these two sides is what is actually projected to the EEG. For it being to the EEG, there are other conditions which are necessary, which we will look at in a second. So if you have a single cell doing this somewhere in the brain, then of course you might not register EEG activity. But if, and that's the bottom line of the next couple of slides, if a lot of cells that all have the same arrangement do this, then this potential difference between somewhere here, relatively far away from the cell, and a reference location will be detectable. And this is what this shows. So again, the same story. You have here now kind of more explicit. You have here a pyramidal cell neuron, which extends all over cortex, has its cell body in layer 5, as discussed. And then you here have some input from some other cortex areas, creating a current sink at this location, and inducing some flow of ions in the extracellular medium that then, in this case, here flows into the direction of a negative pole and has a positive pole here where there's more positivity because stuff is flowing in there. And this kind of configuration of a current sink and a current source somewhere along the cell gives rise to this notion of what is just here shown more abstractly, an electrical dipole, which just means that you have a configuration in space where you have some negative pole and a positive pole, and you just have these two kind of poles, hence dipole, and this being induced on pyramidal cell neurons due to synaptic activity. And the idea is then that this we don't do yet. The idea is that we for something to be detectable at an electrode at in this case actually the cortical surface, it needs to be many cells that are all aligned in the same way and are all temporally synchronously activated, so that this dipole activity or this dipole configuration is strong enough such that the electrical potential it is associated with is still detectable at a site that is a little bit further away from it. In the sense that there's positivity at one point of the cells, and this positivity surplus is still detectable at an electrode that is further away. So the question then always arises, can one really say something about neural activity in terms of these EPRP polarities, so positivity, like we discussed yesterday, the positivities or the negativities, and this is quite hard because these dipoles can be induced in kind of non-identifiable ways by EPSPs and the location of the postsynaptic potential. So this is the example from the Kandel book. So in this case, here you have an electrode site. Here you have some dendrites inducing a positivity. Inducing negativity here, so they basically lead to positivity inflow into the cell here, which means that extracellular gets negative, and you have positivity here. So then at this location where the electrode is, with respect to some reference site, which we just think of as neutral at this point, it gets more positive. Here you have this old convention of plotting negativity upwards. And here you would have some, in this case, some activity, EPSP activity inducing negativity here that then induces the reverse configuration. I'm just looking for. So this would suggest that you can do that, so that you can interpret things in this way around. But what this forgets, and why this is then in the end not working, is that you not only have excitatory postsynaptic potentials as you compare here. So here you have EPSP, and here you have an EPSP. But of course, you also have inhibitory postsynaptic potentials. Meaning that, of course, you can also increase positivity here if these synapses would be IPSPs. So if they would lead to an increased positivity in the extracellular medium here, because it's not an excitatory but an inhibitory synapse, then again you would have a higher positivity here. So this is why people are really reluctant to ascribe any specific neurophysiological meaning to the positivities. I want to go back to the slide because it depends on what kind of postsynaptic potential is, where this is happening, and then it depends on more things, and it depends on where the generating dipole actually is and how it's oriented, and then also positivity and negativity is of course relative always with respect to some reference electrode. And then the question is where the reference electrode is, and hence not too much is made of the positivity or negativity of ERPs. So and these amplitude deflections. So of course for early components that seem to be able to really be reflected by a dipole pattern, it's tempting to, if one fits a dipole to this, and what this means we will see later in the course, it's tempting to try to explain this in terms of, okay, this must be the EPSP activity to the apical dendrites. But then to really know that, one of course would need to record all these neurons and the EEG at the same time to really make that claim. And as long as this is not recorded together, these things could then also lead to the fact that this is actually not the case. What is of course also important in terms of the generation of EEG is that this kind of extracellular currents and potentials being detectable at some distance from where the action actually is. This depends on the spatial configuration, of course, a lot. So, for example, things like nuclei in the midbrain or the brainstem, who are not all oriented in parallel, like the pyramidal cell neurons, they would induce currents and potentials that cancel each other out. So this is what is referred to as closed field configurations, the pyramidal cells that are all aligned and don't cancel each other out are referred to as open field. So this is just this open field configuration. That just means that the dendrites are arranged in parallel, and the current sinks and sources allow the current and hence the potential to spread away from the side where actually things happening. So this is, I think, the bottom line of everything. So which tries to make the connection from again the cellular process. So here you again have the neuron. You have some EPSP activity leading to negativity outside at the apical dendrites and positivity around the cell body. And now the idea is that you have many of these cells, which is shown here. So these, I think you see this. It's not that nice to see. Maybe you see it a little bit better on your screen. So all these triangles here, these are supposed to be pyramidal cells, which extend along the cortex, all having the apical dendrites here. And now the idea being that into one area at the same time there are a lot of EPSPs leading to a negativity at the cortex surface and a positivity at the cortex base or in the white matter, that this then summing over all of this area, that this is detectable at some distance from the from actually this cortex, and leading to the fact that these kind of positivity extend over space in a process called volume conduction, leading to a positivity of the skull at this side of the dipole and negativity of the skull at this side of the dipole. Yeah. So again, I think this is, in a way, this slide, everything that I tried to explain before is again represented on this single slide from the Luck book, where you can of course also read the explanation again. So relating EPSPs at the dendrites to this current sink source configuration with a negative and a positive pole happening at the same time over many of these cells. And hence this negativity, positivity configuration being detectable at some distance and then being projected all over the skull. Yeah, so this is really, and this is already it. So this is the basic model of how EEG activity comes across. It works fairly well, as we will see, for early sensory areas where you have actually things like that. So as I said, for example, primary visual cortex has a big layer 4, so it has a lot of these inputs, so that these pyramidal cells, which are in layer, which are in primary visual cortex, can at the same time have a lot of these EPSPs, which then can nicely project to the EEG signal at the skull. If you're talking about activity later after a stimulus onset that is not that synchronized, and that of course involves many more areas, you still have at the cortex, you have some positivity and negativity, but often it's not that clear cut that you see a dipolar pattern on the skull. We will, on the skull, so on the electrode level. So we will see these dipolar patterns when we then in the next session look at event-related potentials. However, they are mostly detectable and mostly related to early sensory activity. Good. Of course, this is a simplified model. Let me have a look at the time, because I think it should come to an end. This is a simplified model that mainly says that EPSPs and the induced current sink sources in the extracellular medium drive the EEG. But there are also a lot of other processes that have been described in terms of neurophysiology, something like dendritic calcium spikes, burst-induced after-hyperpolarization, direct communication via gap junctions, and glia activity, and all of these, of course, also add into the EEG signal. So this is a kind of simplified model, essentially a lot of research would be necessary if one wants to tease a little bit more apart in how far things like, for example, glia activity can be detected in the EEG. This would, of course, mean that you have to have some kind of preparation where you have glia that adds something maybe to the EEG, and some preparation where you don't have glia. And of course, this, it's kind of demanding, and I'm not really sure that much of this work actually has been done. So there's a simplified story about the EEG signal. This is, however, the working model, and if you look at how EEG data is modeled and analyzed, this dipole model features very prominent in EEG research. So although it's quite simple, and it's actually assumed that in reality things are more complex, it nevertheless works as a working hypothesis and as an explanation for EEG for everything that we do at present. So to see something in the EEG, there need to be many neurons active. They must have a similar spatial orientation, and the neuronal dipoles also must have the similar orientation, right? So it also doesn't happen if you have just the neurons being spatially aligned, but then half of them have an EPSP at the apical dendrite and the other ones have an IPSP, then this will also cancel out. Good. So this essentially try to keep this model in mind, read about it again in the LUC, and then we next time start to discuss what kind of different phenomena actually are observed that relate to this underlying model. That was the question.
Yeah, can you go back to the slide with the general?
This one?
So volume fraction. So yeah, that one. What do we measure? Do we measure the potential differences between one side and the reference point, or the dipole over the skull?
No, what we actually measure is, so we have one electrode. It depends always a little bit. So when you measure it, you have active electrodes. We will come to that at a later point. And then there's another electrode that is somewhere, and that depends a little bit on the system. So some systems have the reference electrode at some point at the skull. Sometimes also the reference electrode is somewhere else. And what then is measured is the potential difference between the electrode here and this reference electrode. Which of course is then mainly a question of polarity. What then happens in data preprocessing, though, is that actually, and this is more the common standard now, that as a reference, the data is re-referenced with respect to the average of all electrodes. And the reason that this is done is that, of course, if you have different reference sites, things can either look positive or negative. And this has been kind of a problem in EEG research. There is a Frontiers article collection about the problem of the reference site. I don't know whether you've seen that. Hence it's generally recommended that one takes, because there is no electrical neutral site, one takes the average of the of all electrodes as the reference. The thing is, the dipole doesn't care how it's measured, right? So the dipole has its negativity here and its positivity here. But if you, of course, have two electrodes next to each other where the negativity is, then they will not have a big difference. If you have an electrode somewhere that is not affected by the dipole, so that's fairly far away, and is constant, relatively constant in terms of its potential, then you will see for this active electrode some negativity. If your reference electrode is on the other side of the dipole, then you essentially see double the difference. So if this just stays at, so this goes negative, so this goes to -10, and here the reference is 0, and you see a -10. If you have your reference where you also have induced positivity, so it's up to 10, and this goes down -10, then you have a difference of 20. So, yeah. The thing is, this is the thing with all these polarities and the relation to cells that is interesting. The problem is, or what you need to do is to really use a biophysically inspired model to make sense of that, and then take these factors of where your reference site is into account. And this is usually not done in cognitive neuroscience. So as I said, DCM for EEG / delay differential equation models, which use kind of Hodgkin-Huxley, Janssen-Witt models for EEG analysis, does that. Standard EEG analysis looks just at the trace of the of each electrode, which goes up and down, and cuts it out around the parasimulus time, and then looks at differences between conditions, but is in no way interested in this. So this is also why there's not much talk about this if you see a cognitive neuroscience talk. this and the other problem is a little bit that these kind of processes that happen at the cellular level and maybe at this column level at the cortex, this is what computational neuroscientists study. So they are interested in how, for example, this cell, if it gets input and projects somewhere else, how another cell then reacts. But they don't. They are not interested in EEG activity. So there are no more insights generated on how EEG activity comes about, because they are happily playing with their different equations that describe these physical activities, and then they might relate it to some cortical or in vitro recordings that somebody else has given them. But it's not trying to establish new principles about how EEG is generated. So from my perspective, there's also, like I said in the beginning, so this model is quite simple, and but there's also a lack of research actually to figure it out in more detail. Good. So then I would stop here with neural origins, and then we continue this course on Friday morning, because tomorrow there's a holiday, public holiday, and this afternoon, as you know, there's the CCNB centennial, so I again recommend going there. I think it's actually for you, it's actually a nice show, because first of all, you see all the people that I just showed you yesterday on slides. Actually, Felix and Timo, they wanted to come, but then they were too busy with all these preparations for this great party that's also happening, that happened yesterday. So you see all of them, and you also hear about the research that has been done at CCNB, and in the end there's this public part, which I think also might be nice for you if you need additional motivation for studying what you're currently studying, because that's one thing. Before we close, I want to say, so I don't, so as you might have noticed yesterday, I don't stand here and say, oh, it's also amazing and we can study the brain, and this is also like, it's the most important thing in the world. And of course, so the reason why I don't do this is because I assume that we all agree on that already. So that I don't feel that I have to motivate you, and I will not motivate you by telling you, by understanding the brain, we understand ourselves, and so I believe all of that, and this is why I'm doing all of this. But I assume that you also already believe all of that, and hence are here and motivated to study that. However, if you feel you need another motivational push to get started on the semester, I think also this public part today is nice, because it will be all how amazing this is, and Rainer Göbel will tell you what amazing things we can do with fMRI. And so if you feel you need another motivational push, you will get it there. You will not get it from me. I will always say it's really complicated, it's really a lot of work. But that's also how it is. Good, then see you around.